It's a real pleasure to be here today. I have titled my talk 40 Years of Success, which I really feel standing before you today. I'm lucky enough to have followed Pat's excellent talk this morning, and I have to say I think Pat had the raw end of the deal there. You pulled together our history so beautifully, so much complex history. Everyone I've spoken to, I think, is just quite amazed at how much research has come out of this fantastic resource. I get to be a little more whimsical. I get to make my personal reflections because I'm also celebrating 20 years last month with this fantastic resource. So the Brain Bank, supported by Brace and myself as a Brace-funded staff member, have sort of grown up together in that respect. I'm going to take you slightly back to the beginning for those who've just joined us. Pat gave us a bit of a refresh, but the Brain Bank was originally established in 1984 by Professor Gordon Wilcock, with Brace being founded slightly later in 1987. I have to say thank you to Gordon, to Seth, who's in the audience, and to Pat for giving a very young, very inexperienced me the opportunity to have this fantastic career and to help to build this resource into what it was today. It was quite a formidable interview panel at the time, but I've reflected on how much I've learnt from those friendly and amazing faces. And I have to say, I wouldn't be where I am today without them. The Brain Bank was originally housed in the John James Laboratories, founded by the John James Foundation Trust at French Hospital, and that was under the Department of Care of the Elderly. It later became associated with the Dementia Research Group, which Pat and Seth headed up for many years, and now Pat and Dr. Scott Miners in the Bristol Medical School at the University of Bristol. It was quite interesting when I was reflecting on what I was going to talk to you about today, that the mission statement, the overall tagline of what we wanted to do as a resource really hasn't actually changed that much, with our mission statement today being to support dementia research that has the potential to contribute to our understanding of the underlying diseases or to be of value to those affected and their families, something which I hope I'll show you that I think we really are living up to. I'm taking you on a trip down memory lane, so I wanted to share some of the older photos. I'm delighted that Sonia Matthews is here in the audience with us. Sonia is my predecessor, if that's the right word to use, and a hard act to follow. And some of the other faces from the very earliest days of the Brain Bank shown here. I'm loving your hairstyle. Sonia, we've decided you don't look any different at all. I think what I wanted to show you with this is, and what I hope will be a reflection as I go through my talk to you today, is just how many people have been involved in the success of this resource. This was built, these foundations were built by these people here. And what a resource we have. So Sonia was very much a one-woman band. I'm very lucky to have a fabulous team who are all here behind me. And actually, I'm really more of a figurehead nowadays, and they do a lot more of the hard work. So thank you very much to them. So where are we today? We're in this fantastic, modern, newly built extension in the Learning and Research Building. Pat reflected earlier, we've also been in there 10 years, which is quite staggering, really, for those of us that remember the stress of the move. We are, I would argue, a world-leading research resource of post-mortem brain tissue and cerebrospinal fluid, both from people with dementia or other neurological conditions, and also particularly those who haven't been affected by neurological conditions, so people that we use as controls for experimentation. We're licensed by the Human Tissue Act, which was introduced in 2004, but actually came into force in 2006, with Pat being instrumental, being named the designated individual for that licence, which I have had the pleasure, should I say, of taking over since. One of the things that I always reflect on is, although the Human Tissue Act was introduced because of less than ethical practice, Gordon, Seth, Pat, the earliest people involved in our Brain Bank had very ethical processes, and they were ethical before they had to be. And I think that's something that's really important in our history. We're an NHS Research Ethics Committee approved research tissue bank. A lot of words, but that basically means that we can supply human tissue for research to researchers in the UK. It's enabling research to be conducted more easily rather than researchers having to go and apply for their own ethics. That's particularly important when we're able to help fund some pilot work to get those earliest ideas started and to enable those ideas to come to fruition. This is where we are today with the current Brain Bank team. Obviously, Pat, Professor now, Liz Coulthard, who are now our co-directors, myself and Professor Johannes Atoms from Newcastle University, who's acting as our interim pathologist following Seth Love's retirement. I have to say he's irreplaceable, but we're doing our best. He's pulling a face at me. We also have Candida, Taya, Kath, Gideon, Jo and Meredith. I've also failed to put on there, because she's so new, our new part-time brace-funded administrator, Dill, who I'm very happy to have come into our life to help us move our paperwork along slightly more efficiently. But I think it's really important to show you how the team has grown in these 40 years. We couldn't possibly do with less staff members than this at the moment. And in fact, we are all stretched and working to capacity. That can only be a good thing. I'm going to talk a little bit about our uniqueness, what makes us different, why we're so important. Many of you may know that we're one of 10 brain banks in the UK that make up the UK Brain Bank network. We accept brain donations from the whole of the southwest of England, but I've put and beyond, because where possible, we will accept donations from as far afield as we can if someone really, really wants to donate and it isn't possible through other resources. Although there are 10 brain banks in the UK, there's been a huge shift in the landscape of funding for brain banks, which unfortunately means so we're only one of 6 who are actively supplying tissue for dementia research in the UK. There was a downgrading of operations in October 2021, where there was the withdrawal of some of the funding of other banks in the network. I have to say, thanks to Brace's support and that of Alzheimer's Society and Alzheimer's Research UK, we have fared much, much better than others. And I think we're actually in a very strong position now with that backing that we have. However, that contraction of the network means that what we're doing here in Bristol is more important than ever. We're meeting the needs of researchers where perhaps others are now less able to. We're also involved in a national project called Brains for Dementia Research. We joined BDR in 2010. And we were slightly latecomers to the party for various reasons, but I think we've more than made up for it since. BDR across the UK is now a gold standard for brain banking. So it involves 5 leading brain banks, of which we are one, and a research centre in Cardiff. Together, we work as a network of using common standards, best practice and cooperation. And that means that researchers can pull together samples from all of these 5 different brain banks and know that all of the tissue will have been donated, treated in the same way, characterised in the same way. And it's enabling much, much larger populations for researchers to use, which is resulting in really good quality research. We currently have just over 230 living participants for BDR in Bristol, and we assess them on an annual basis. And by we, I mean Catherine, our research nurse who's with us today. We've successfully received 175 of these BDR brain donations since we joined the project. And we've also just undergone a reconsent process for a number of the Cardiff donors, meaning that we now have 100 extra brain donations from that cohort who will be coming to us in Bristol. That means that we're going to be the largest participating brain bank in this national project. And that puts us in a really good position in terms of supporting quality dementia research going forward. You may ask after 40 years, why do we still need a brain bank? Why do we need the South West Dementia Brain Bank? What are we doing with donated tissue? My answer to that is always neuropathological diagnosis. The only definite way to diagnose dementia and the type of dementia is to examine post-mortem brain tissue after somebody dies. So we both collect and provide that tissue to researchers, as I'll show you later, but we also enable collaboration between groups of individuals who perhaps might not think about working together, so clinicians and basic scientists or animal scientists, and we bring them all together to make larger, bigger, better projects. We maintain standards of excellence in tissue banking. As I said, I'm now the designated individual for our human tissue licence, and that's something that's particularly important to me, is making sure that our donors' tissue is used in the way that they wanted, it's stored correctly, it's treated properly, and with the respect and dignity that our donors and their family members deserve. We have so much to learn about common diseases, Alzheimer's, all the hundreds of different types of dementia, as well as the rarer or emerging disorders and new pathologies that we see as techniques and laboratory processes move forward. So comparison of brain tissue, as you can see on the screen, on the left-hand side we have tissue from someone with no known neurological condition, and on the right from someone with end-stage Alzheimer's disease. Making comparisons between these brains, this brain tissue, is crucial for us to find out why these different types of dementia occur, how they differ, how they progress. And as Pat's talk excellently showed us this morning, for those who weren't there, how we might target treatment strategies of the future. How are we going to prevent these conditions and how will we cure them? It's difficult, impossible at the moment really, to develop a good model of a human brain in the lab. So animal models do give us a good insight, but there really is no substitute for the real thing. So all of these different parts must play their part to give us the larger picture, which is then translatable into humans, people living with the condition today. Human brain tissue has been critical to nearly all major advances in dementia research. So that's why we still need the brain bank. Clinical diagnostic accuracy. We use Alzheimer's disease here as the example because it's the most common type of dementia. But although this review is quite old now, so an NIA review, 2013, of the accuracy of the clinical diagnosis of diagnosing Alzheimer's disease showed that about 25% of patients with AD had been misdiagnosed as having something else, and about 40% of patients who had something else had been misdiagnosed as having AD. Those statistics are just quite staggering to me still, I have to say, and it is truly reflected in the cases, the donations that we see in the brain bank. So approximately 40% of the brain donations that we receive, once we have that end neuropathological diagnosis, that diagnosis either doesn't meet the diagnosis that the patient was given in life, or may have multiple different pathologies which have confounded this, this picture, this clinical picture. And when we talk about the clinical trials that are undergoing, that are given for people with dementia, specific dementias like Alzheimer's disease, if you were to reflect back on that and think that possibly 40% of your population that you've been giving these drugs to may not have the condition you're trying to treat, or may have other pathologies which may affect the action of those treatments, we can see why we're here today without new treatments for these conditions. So brain banking has a really important role to play in increasing the accuracy of diagnosis, in allowing our clinicians new tools or development of new technologies to increase the accuracy of clinical diagnosis for those living with the condition. I always add a caveat to this. Our clinicians are doing a fantastic job in the NHS and beyond with the tools that they have available into making these diagnoses, but as we still know, we do find that that varies. We're very lucky here in Bristol to have the memory clinic and Professor Liz Coulthard, who is leading the way in clinical diagnosis. As I said, this is slightly self-indulgent, so I'm going to talk about subjects which I feel are important. In my 20 years at the Brain Bank, lots of people seem to think that my job is about death. And yes, sadly, we do deal with a lot of death. I always bring it down to, I feel that the majority of what we do is actually for the living. It's for those who are left behind. It's dealing with people. It's for dealing with those people who are living with the condition who will come after them. A fantastic nurse once said to me that people only die once, and it seems very silly, but it's something that I've taken with me for my whole career. What we do at the Brain Bank, we have one opportunity to make the donation experience, to make it a positive experience, for people to take something out of it, to feel that they and their loved one has done something which is going to help people going forwards. And I think that's one of the really important things we do. And it isn't something that you can write an impact report on. You can't generate figures on it. It's just about the personal experience and the people and the families that we've dealt with over this time. I've mentioned the neuropathological diagnosis. So within 6 months of receiving a donation, we provide this neuropathological diagnosis, so the definite diagnosis, to the donor's family, loved one, but also to the GP, a consultant, if one was involved in the person's care, and the assisting mortuary. That's really important in increasing the accuracy of diagnosis. So we've worked very closely with Liz's team and clinical teams to make sure that if a donor's diagnosis was incorrect or differed, that we're able to feed that correct diagnosis back to them and offer learning and teaching opportunities. They may be able to go back and look at cases together to find where symptoms missed. Was there a different test that we could have done? And that may change that diagnosis process for someone going forward. It's only once that diagnosis has been made that the brain tissue becomes available for distribution to researchers. And I think that's something that people forget. They think once that donation has been made, that that's sort of the end of the road. Really, that diagnosis is actually the beginning of the road for that donated tissue. Tissue stored may potentially be used in hundreds of projects over decades. So incredibly, we have some of the earliest donated tissue still in storage in our brain bank, which could still be used in research today. I don't want you to take too much of this in. It isn't intended to scare you. This is intended to show you how we differ from other brain banks, or what a fantastic resource we have here. We hold tissue from over 1,340 donors, and that tissue is stored in a variety of formats so we can make it available for different types of research. But we also have this wealth of clinical data, and this is something, I'm showing you an extract of the UK Brain Bank Network database, which Seth was absolutely instrumental in developing into the tool that it is today for researchers and for brain banks actually. You can use this database to do incredibly complex searches, both for tissue, but also for clinical data. And that's one of the things that has changed dramatically since Sonia's day, since my earliest days. When we would receive a brain donation, we would actually we would know so little about the individual. We might know their sex, we might know their age, we would probably know their cause of death, although not always in some of those cases. And that was about it for a while. We know now that for research, there are so many things that influence how this person ended up with us, how they ended up with this condition, what may have developed that condition, any comorbidities, things like type 2 diabetes, hypertension, all of these risk factors that we now know are potentially modifiable for changing the course of both developing dementia, but also dementia progression. All of this clinical information is tied together in this database. And a project that's been really big, something I've wanted to achieve in over a decade at the Brain Bank, was the digitisation or the scanning of all of our donors' medical records. And that's something which we were able to complete as a project. I see Sonia raising an eyebrow. I know, you never thought it would happen, did you? So one of the important things we do when we receive a donation is we apply for a donor's medical records from birth to death. We want to know everything we can about these individuals. That has only ever been in a paper format. And if you've ever tried to read GP medical records from 60 years ago, I can tell you it's not fun. So we've digitised these records, and although we still can't read their writing from 60 years ago, what we can do is anything that was previously in a text format is now in a text searchable format. So we can go and interpret these records for key information, individual drugs, blood pressure readings. We can look for things like people's BMI. The list is really endless. And when you add that to the highly characterised, excellent quality tissue that's available, you suddenly have an incredibly, incredibly powerful research resource. So we're the first brain bank in the UK who's managed to complete this exercise in full, and that's something that I'm incredibly proud of. So basically each individual case has this wealth of information. We have their clinical history, we have this detailed neuropathological history, we also have some biochemical, some genetic data, we look for the ApoE genotype of all of our donors as well, which we can tie in together and make available to researchers. As part of our work with the Brains for Dementia Research Project, we also have a subset of bloods taken from our living participants, which are then available to go with their brain tissue after these participants die. That, and there's a new project looking at the generation of cell lines from donated brain tissue. And so there's also a subset of those cell lines available to researchers. And they're quite unique because they have both a clinical diagnosis and a neuropathological diagnosis. So that's something that we haven't necessarily seen or been able to access before as researchers. I hope you're getting the picture that we have done huge amounts, excuse me, in the last 40 years to increase our understanding of dementia. So we still have around 750 potential donors, participants who are registered with us who want to donate their brain after they die. We receive around 40 to 50 new brain donations per year, and that's really important. You may think, well, we've had over 1,300 donations, why do we need any more? If we're doing our job as a brain bank, we shouldn't be holding this tissue. We should be bringing it in, we should be characterising it, and we should be sending it out. Otherwise, we're not creating a resource that is of use to researchers. This is a new figure I generated just today, and even I was quite shocked. So I have really detailed records going back to about 2011. In that time, we at the Brain Bank have provided it's 81,577 individual samples to researchers. I mean, that figure is incredible. It nearly rivals the number of samples that we have still stored in the Brain Bank. So these are huge numbers from what is actually quite a small team, is a charity-funded resource here in Bristol. And those samples alone have underpinned 100 scientific publications in peer-reviewed journals that have all improved our understanding of these conditions. And I think I suddenly felt a huge pride in my role and that of my team. I think when people, so I was lucky enough to do a Brace-funded PhD. Brace really have supported my career throughout. And when I was finishing up my PhD, everyone was saying to me, what do you want to do? Are you going to become a brilliant academic researcher? Will you make professor? And I said, absolutely not. For those of you who know me personally, I'm not the greatest academic mind, but what I am is really, really passionate about dementia research. And I feel that myself and the team have underpinned more research here than I could ever have conducted in my entire career as a research scientist. So that's something I'm really, really proud of for all of us. I wanted to highlight some successes to you. I'm not going to read these verbatim, but I really want to highlight the differences that can be made. We've talked about all those hundreds of publications, and these are some local ones. So these involve the Dementia Research Group. I have to say, I'm pretty sure that everything I'm talking about involves Pat and Seth, who have just led on such a huge amount of this research that has come out of Bristol. So we've been lucky enough to be involved in some very high impact publications. The one I highlight here shows that infections such as urinary tract infections and bronchopneumonia worsen the cerebral blood flow and the function of the blood-brain barrier in people with Alzheimer's disease. And we certainly know that from looking at brain tissue. So around 50% of the people who have a diagnosis of Alzheimer's disease also have some cerebral vascular changes within their brain. Pat mentioned COVID earlier, and I'm going to mention it again with Dr. Lindsay Sinclair, who is a psychiatrist working with the Dementia Research Group. And she looked at the dementia incidence rates before COVID-19 lockdown and during and after. And basically that showed that while the protective measures used by the UK government didn't necessarily lead to an increase in dementia, that the measures may have led to a more rapid deterioration in people with the condition. And I think I can see people nodding. I think that's something that we all felt. COVID-19 and the various lockdowns were not good for people with this condition. Collaboratively, and there has been lots and lots of collaborative work, researchers have shown that most people with dementia have multiple concomitant pathologies. I think that's something I've certainly taken away in the last decade. There's been some new and emerging pathologies which I'd never previously heard of, and we now know that people do often, I'd say more often than not, have these multiple pathologies, and that's really important when we're looking at potential treatments or preventions. We need to know and unpick these pathologies to make sure that we can treat people. Maybe that someone with pure Alzheimer's disease needs a different treatment to somebody who has a different pathology. DNA from our cases has contributed to the largest genetic study of vascular cognitive impairment, and that's incredible, and that was led by Pat in collaboration with the European Alzheimer's DNA Bank. So further successes, developing guidelines. Again, I feel like I've highlighting some of what you've said, but I think it's important what we've both chosen to reflect on. So the bank and the staff in the bank and those of the Dementia Research Group have played a vital role in increasing our understanding of the role of cerebrovascular disease, both in dementia and in particular in Alzheimer's disease. Doctors Fisher and Miners are developing new approaches to characterising blood vessels within the brain. So the donated brain tissue can be used to develop new techniques, and this is a really good example of that. They're now able to visualise the anatomy of blood vessels in 3D, which will enable them to characterise them in a way that just hasn't been possible before. And so these are techniques that other groups and other studies will pick up on and will develop. I mentioned Lindsay again because in the next example, because Lindsay is slightly different in that she is a psychiatrist, but she's done some work using the BDR data. So this is where this clinical data is really important. She's published work looking at later life versus early life depression and showed that individuals over the age of 50 with depression didn't have any evidence of increased AD pathology. But this is in direct contrast to the vascular depression hypothesis that has actually been prevalent in her field for a long time. It also demonstrates that although depression is a risk factor for dementia rather than an early symptom, at least in individuals with severe depression. So this is really important from a psychiatric point of view in terms of how we deal with all people, older people with depression, and whether or not hypothesising whether or not these actually might be slightly different conditions and might need to be treated in different ways. The last example is really just a small one, but we have been involved in the analysis of past clinical trials. So this was one that was looking at the peptide A-beta42, where people were immunised with this in an attempt to clear the beta amyloid from the brain to see whether or not it made any difference. We had a very small part in this, but I would really like to see this in the future of brain banking. I would love us to be in a position where we were able, as part of the clinical trial model, to ask people at the time, would you be willing to donate your brain? To have the ability to go back later to perhaps reanalyse some of the data, to group these people into slightly different groups. Did they have AD? Did they have AD with vascular changes? Maybe they had some other pathology. I do feel that we would really see perhaps some very different results, and we may have missed some very important drugs in the area. This is more for fun because I'm going to lighten the load. I was so proud to be a Brace-funded PhD student. I was lucky enough to do my PhD under Pat and Seth, who were very patient and very amazing. I struggled hugely at university, and I only obtained a 2-2 in my degree, which meant I needed a special letter from these pair to allow me to progress to PhD. I hope you don't have any regrets, but you can't have it back. Brace was a huge part of that. I didn't come from a background where I could have done a PhD without funding, without the support of so many people. It has changed my life. It really has. And I'm so proud to have had this opportunity to be able to reflect on this with you today. And I just wanted to share some of the fun memories. You know, a lot of what we do is very serious. It's very important. But we have this wonderful team. We have this wonderful, all these wonderful people around us. And I like to think I've done a little bit to try to pay Brace back over the years. An example, this is me abseiling down the Avon Gorge for Brace. I did also run half marathon, Bath half marathon, but the pictures of that weren't so pretty, so I haven't included those. But we've had a fantastic team around us, we really have. And for that, I'm incredibly, incredibly grateful. The years ahead, I think we should be looking forward. We're looking back, we're reflecting on where we are. It's really important to look forward. I hope just this short segment today has cemented for you exactly why the brain bank is so important, why deceased human brain tissue matters to the living. It matters to people going forward. The clinical trials that we're going to need to develop potentially prevention, but hopefully cure of these conditions relies on human brain tissue. We need this resource to be able to build the picture, to build that understanding, to translate into humans. We also are lucky enough to have this fantastic community around us. We have this huge, over 700 people who are highly committed participants. They want to take part, they want to make a difference, both during their lifetimes and after they die. We have this availability of the extensive clinical data that we're working on expanding more and more. And we also have an increasing number of brain donations with this linked data. So both standardised assessments in life, standardised assessments in death, which is particularly vital to dementia research. So it really just remains for me to say thank you. Our sincere thanks to our supporters, and that's all of you. That's Brace supporters, that's staff members, that's researchers. You're all our supporters over the years, but in particular, our funders, of course, to Brace, Alzheimer's Research UK and Alzheimer's Society. But most importantly, it's really nice for me to stand here today to thank our donors and their loved ones who contribute everything. Without them, none of this research is possible. And with that in mind, I'm just going to read my own quote. I put it on the screen because I knew I'd forget it. But brain donation is a truly altruistic gift. I've been privileged enough to oversee over 600 successful brain donations during my time with the bank. Each one of these individuals wanted to change the course of this devastating disease for those who came after them. And I see that hope realised every day in the research that has been undertaken with their tissue and the knowledge that has been gained from it. I'm going to leave you with this final slide, which is just a bit of a compilation. I can't show you the faces of our donors. I can't show you the faces of their families, but I can show you just a few of the fantastic people who have built, who have been part of, created this fantastic resource that we have and we are able to use today. So thank you very much.